Hello, welcome to the next in a series of review clips designed to assist you in preparation for your Biology 321 public examination. In this clip, we'll be taking a look at protein synthesis. Protein synthesis is the process whereby the instructions from DNA are used to create polypeptides that make up a protein. This process is also known as gene expression. Gene expression, by definition, is the transfer of genetic information from DNA to a protein. And a polypeptide is a long chain of amino acids. Two or more polypeptides joined together is the same as a protein. So why do we even bother making proteins? What difference does it make if they're there or not? The reality is that proteins certainly are the important structural components of cells and your body as a whole. Proteins are what make you operate. For example, proteins are found in skin, hair, in our blood, hemoglobin, by way of one example. Enzymes and hormones are all comprised of proteins. Our DNA determines the sequence of amino acids in the polypeptide. This is determined by the sequence of the nucleotides contained in the DNA. Protein synthesis as a process occurs in two steps. Initially, there is transcription, and transcription is the process whereby DNA is converted into messenger RNA. And following transcription, there is translation. Translation is the process of translating DNA into a protein. Transcription occurs within the nucleus of eukaryotic cells and in the cytoplasm of prokaryotic cells. It occurs according to the following steps. Initially, RNA polymerase will attach to the DNA molecule inside the nucleus. Only one of the strands of the DNA will be used to make the messenger RNA. This strand is referred to as the antisense strand. The other strand is called the sense strand. It's not used. RNA polymerase will attach to the antisense strand and open it up. It then begins reading the strand from the 3' to the 5' end, laying down RNA nucleotides as it moves along. As the RNA polymerase passes a region on the DNA molecule, the DNA will then zip back up, and the messenger RNA will in turn separate from the antisense strand. When the messenger RNA strand is complete, it contains exons, that being the coding regions, and introns, or non-coding regions. The introns are removed and the exons are joined together. This is done by another series of enzymes. The completed messenger RNA molecule, which is of course single-stranded, an RNA sense strand if you will now leave the nucleus and enter the cytoplasm. This concludes transcription. Once the messenger RNA is rendered to the cytoplasm, it is then ready to begin the second phase of protein synthesis, translation. Translation occurs within the cytoplasm of all cells according to steps you see listed. Initially, messenger RNA will bind to an active ribosome found in the nucleus. The binding of the messenger RNA happens such that the first two codons are exposed. And all a codon is a sequence of messenger RNA molecules, 3 of them, that code for a specific amino acid. For example, the sequence AUG would represent one codon. The sequence CCC would represent another. 3 nucleotide bases comprise a codon. A transfer RNA molecule then, carrying amino acid base pairs with the first exposed messenger RNA codon. This happens when the anticodon on the transfer RNA recognizes its complement on the messenger RNA. And by definition, an anticodon is a sequence of 3 bases on the end of a transfer RNA molecule that recognize the codons of messenger RNA. A second transfer RNA molecule will then carry an amino acid arrives at the codon adjacent to the first one. Enzymes cause the amino acids to join together in a peptide bond. The polypeptide gene is then transferred from the first transfer RNA to the second one. The ribosome, while this moves down one codon along the messenger RNA strand. The initial transfer RNA molecule leaves to go get another amino acid, and then a third arrives at the exposed end to the next one, and the process repeats. This process continues until a stop codon is reached. At this point, the polypeptide is released from the ribosome. A stop codon is a sequence of 3 bases which indicate it is time for the protein to stop building. So we talked about transfer RNA. Transfer RNA is a cloverleaf-shaped RNA molecule that's found in the cytoplasm cells, and it's called transfer RNA because it brings amino acids to the ribosome and recognizes the codons on the messenger RNA, so it can find the correct sequence of amino acids when making a polypeptide. To diagram it, a transfer RNA molecule looks like this. It is cloverleaf shaped, it's comprised of bases. There are 2 ends of significance. There is an amino acid attachment site where it attaches a specific amino acid. On the opposite end, there is an anticodon. The anticodon codes to a specific region of the messenger RNA based on its complementary base pair. In completing problems, completing questions on your test, you will need to read a codon table. All a codon table is a table that lists a series of messenger RNA codons that are read by the complementary transfer RNA molecule to determine the sequence of amino acids. For example, each codon is comprised of 3 bases. So if we knew that the DNA sequence was A, T, C, the corresponding messenger RNA would be U, A, G. To determine what amino acid would be brought by its transfer RNA complement, you would read U is the first base, A is the second, G is the third. This would be a stop codon. This would mean that at this point, no further amino acids would be joined. If the DNA sequence was A, 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 the messenger RNA sequence would be U, U, U. To read the table, the first base is U, the second base is U, and the third base is U. This codon would code for phenylalanine. So in reading a codon table, you read the following step. Initially, convert DNA to RNA. Using the 3 letters from the RNA codon, look them up in the table. Work from left to right using all 3 letters. In the table, the word first stands for the first letter, second for second, and so on. When you have read all 3 letters, you will arrive at the name of a specific amino acid. And you will continue to do this for each sequence of bases from left to right, or the 5' to 3' direction. For example, AGG translates to UCC. First letter U in the top row, second letter C, second column first row, and third letter C. This leads us to serine as the correct amino acid. The codon table is for messenger RNA only. You will need to convert from DNA to messenger RNA, or possibly from transfer RNA back to messenger RNA, depending on the nature of the question. In the past, public examinations have included questions on this topic, and I present a few of them for your consideration here. This question asks you to refer to the diagram that you see in front of you to answer a couple of questions. The first question asks you, which molecule serves as a template for the production of structure D? The first step in this question is that you need to recognize that structure D would be messenger RNA. You are asked to then determine what molecule serves as a template for the production of messenger RNA. That answer would be C, DNA. You are asked what type of structure is partially formed by molecules E, F, and G. To answer this question, you would need to recognize that E, F, and G refer to amino acids. The structure is partially formed by molecules E, F, and G would be a polypeptide B. This question, as posed, asks you to refer to a codon table. You are asked which DNA sequence was used as a template to produce the polypeptide sequence glycine, isoleucine, phenylalanine. Take note, you are asked about DNA. Therefore, you will need to determine the messenger RNA sequence, and from that, determine the DNA. The first amino acid, glycine. I'll abbreviate GLY. The possible codons for glycine, as you see from the table, glycine is located here. The possible codons are GGU, GGC, GGA, or GGG. The possible DNA from this would be, writing this time in green, GGU would become CCA. GGC would be CCG. GGA would be CCT. And GGG would be CCC. Looking at your possible choices, CCC, this is a possibility. BCCG as well is a possibility. GGA, while this would be the messenger RNA, it is not a possibility for the DNA. And DCGGC, again, well, this might very well be a possibility for the DNA. It is not a possibility for the DNA. So we can eliminate that one as well. From here, we now move to the next amino acid in sequence. In this case, isoleucine. Isoleucine, which is located in your table here, has as a possibility for RNA A, A-U-U, A-A-U, A-U-C, A-U-A. The possible DNA sequences from this would be, again, DNA would be T-T-A, T-A-G, or T-A-T. T-A-G is a possibility. T-A-A is not. The correct answer then is going to be A, C-C-C, T-A-G, A-A-C. This question asks you to identify the process that is occurring in the diagram below. If you recognize that what's happening is a messenger RNA strand is being formed from a DNA template, this question is relatively easy. This process is, of course, transcription. For 3%, the following constructive response question was posed. A mutation changed the DNA template sequence from AGC, CCG, GCA, TTG to AGC, CCG, GGC, ATTG. You were asked to identify the type of gene mutation and to use the codon table to show how the polypeptide sequence changes from the original to the mutated strand. I would ask that you press pause now in your learning clip before concluding. Attempt this question before looking at the answer key. The answer for this question, for 1 mark, required you identify the mutation as being an insertion. A base was inserted between positions bases 7 and 8. And through the use of the codon table and deciphering both strands, you would have been able to determine that arginine was changed to proline for 1 mark and asparagine was changed to a stop codon for another mark. Thank you for your patience. I hope you found this learning clip to be helpful.